student, Matt Laster, came to my lab in 2008, and he had noticed even before arrival that in conventional T1 weighted images, this is place, that in the cortical gray matter ribbon, some regions are a little brighter than average, other regions are a little darker than average. And then if you take a T2 weighted image, different pulse sequences giving a different pattern that's essentially the mirror image of contrast. In the gray matter and a T2 weighted image, the patches that had been brighter are now darker than average, the ones that had been darker are now brighter than average. So Matt cleverly did the math, took the ratio of T1 versus T2 weighted values for each voxel or volume element, colorized it to make it more attractive, and then mapped it accurately to the cortical surface to generate maps that turn out to be strikingly well related to maps of myelin content from postmortem analyses. And so we call these myelin maps, and the red represents heavily myelinated regions, the blue and indigo represent lightly myelinated regions, and other colors in between. If we do this for hundreds of subjects and align them very accurately in ways that I don't have time to detail but are very important, we get very crisp-looking myelin maps on the left here. And if we look at the cortical thickness using the pre-surfer models that I mentioned before, we get a thickness map on the right. And in both cases, we can see features of systematic variations in myelin content or thickness that are strongly related to areal boundaries. We call these areal features, and we use spatial gradients, what we call gradient ridges, to look at evidence for areal boundaries, which are very informative for some regions, but in prefrontal and other regions, the lightly myelinated regions that we are associating with cognitive processing regions, the structural measures of T1 and T2 aren't so informative, so we draw on other modalities, particularly functional MRI. So for that, Deanna Bart spearheaded the effort to get high-quality task MRI data, and we got an hour's worth of data from each participant using 7 different tasks run in sequence. I'm going to quickly illustrate 3 of them. One of them is a language task, actually listening to stories in the scanner versus doing arithmetic in the scanner. Another is what we call a social cognition task, so looking at these simple images and asking, are these images, these triangles, just moving randomly past one another, or are they interacting in a social-ish way, hugging and kissing and dancing in ways that make a big difference inside the brain? And the third exemplar task is what we call working memory. So is the image one is looking at right now the same or different from the one two previously, a so-called two-back process where we had different types of images to be help us in analyzing the data. So we can look at this task MRI data mapped onto cortical surface models that are inflated, as you see here on the left, on subcortical slices, and also on a cerebellar flat map shown in the lower right. So just to give you a very quick glimpse at what happens inside your brain or HCP subject's brain during these kinds of tasks, when listening to stories versus the baseline contrast, that lights up in yellow and red patches of cortex, swaths of cortex in lateral temporal, prefrontal regions in different patches in both the left and right hemispheres, although it's stronger on the left as expected. If we take the next task, the social versus random, we see a strikingly different pattern. There's some overlap, but mostly different regions, and the quality of the alignment lets us separate regions that would have been blurred by conventional analysis and visualization methods. And the third task is the working memory, which lights up yet a different region, or set of regions, particularly in lateral parietal and prefrontal cortex. So just to summarize quickly, each task that one does in a scanner engages a complex network of brain regions that overlap to some degree. They're typically not totally isolated, but they're rich in information of spatial organization. If we go to a different modality, again using functional MRI, but now using what we call resting state, the lying quietly in the scanner, staying awake and thinking about whatever, what's for dinner, who's going to win the basketball playoffs, etc. That leads to a complex symphony of fluctuations in space and time across different brain regions, and one can analyze that by taking any pair of gray matter locations in the cortical or subcortical regions, looking at their functional MRI time course after cleaning out the noise that complicates the issue, and ask, are the patterns correlated perfectly or imperfectly, or are they even anti-correlated? And that leads to all pairwise comparisons of correlation, giving us what we call a dense functional connectivity matrix, or a dense connectome, shown by this matrix over here, and we can sample that one location at a time, take one region, for instance, in lateral parietal cortex on the right hemisphere, and ask who's correlated with that along that row, and one sees a functional connectivity map for this exemplar seed in parietal cortex, a totally different pattern from a central sulcus pattern, and this is very informative, and it's led to thousands of studies around the scientific literature, but it raises a fundamentally important question: to what extent do these functional correlations that we label as functional connectivity, how much do they actually relate to anatomical connectivity? That's currently essentially impossible to address incisively in humans, but we can turn our attention to animal models. So why do we care about this? Brain circuitry mediates behavior, as I already argued. For human cortex and brain, our ability to infer connectivity is related to 2 different modalities: functional connectivity that I've introduced and we'll talk more about. I'll just mention that what we call structural connectivity uses a completely different modality, diffusion imaging, and an analysis that we call tractography to infer estimates of connectivity. But both of these methods are highly indirect. They have serious limitations that are progressively being better appreciated, but we care about this not only because of our fascination about brain circuits, but also because many brain disorders are hypothesized to be abnormalities in connectivity, a so-called connect subtopathy. And if we we want to get a better handle on this, we need better analysis, critical assessment of whether these methods actually yield robust insights about real brain circuits. And to do that, we really need animal models. So that's part of why I introduced the macaque and marmoset animal models as substrate for getting accurate anatomical connectivity data. So to bring us back into macaque connectivity, this is an example of a retrograde tracer injected into one brain region, and all of the retrograde labeled cells are shown on this for this particular slice in little patches of gray. If we take this slice and just look at the contours, we can identify on our MRI-based macaque atlas a corresponding contour, and we can do that for every slice that has tracer data in it, and use that strategy to map the distribution of connectivity from a single tracer injection, in this case in ventromedial prefrontal cortex, throughout the entire hemisphere, whether it's weak or strong. And we can do that for another injection in a different brain region, still another injection. So far we've done that on 28 different example injections, but we're continuing to do this work with Henry Kennedy's lab to get accurate maps of real connectivity. Then in a parallel collaboration with Takuya Hayashi's lab in Japan, we're looking at functional connectivity from a different set of macaque monkeys, but using high-quality data acquisition and analysis. Takuya has been able to analyze 30 different anesthetized macaques and to generate analyses of seed-based functional connectivity using resting state fMRI. So here's a seed in one region of medial prefrontal cortex, and you can see a pattern of functional connectivity on the inflated map and on a flat map. And if we move the seed location a little bit, you see a totally different pattern showing that this method is informative. But we can now compare anatomical and functional connectivity using matched seeds. So here's an anatomical tracer injection in a premotor area F4, and here's the functional connectivity, which shows many similarities but many differences. And a different seed location in prefrontal cortex shows a different anatomical connectivity, and in the middle panel here, a different functional connectivity. They are correlated but far from perfectly. And if we do this quantitatively and ask what is the correlation between each tracer injection anatomical connectivity and functional connectivity, we get values where the correlation using what we call full correlation, that's most straightforward analysis, is up to 0.6 or more, so not fantastic but pretty good, and it falls off for different areas. And one can do different analysis methods, one called partial correlation that I'll not try to explain in detail, and get different estimates. So the point at this stage of the exercise is to say we're making headway in quantifying the degree to which anatomical and functional connectivity relate to one another, but we have a long ways to go, but we're getting more data and have different analysis strategies to bring to bear. So with that as background, let's now move on to human cortical parcellation, which I've introduced previously by showing you at the very beginning the Rosemont cytoarchitectonic map, the Votes much more fine-grained myeloarchitectonic map. In recent years, a number of investigators have used fMRI, particularly resting state fMRI that we've already talked about, to estimate cortical organization and to parcellate the cerebral cortex, as shown here for Randy Buckner's and Thomas Yo's parcellation. As one example, Evan Gordon and Steve Peterson's lab came up with a different parcellation that has more than 400 parcels, but it has issues with it. One is that the map is incomplete. There are some regions that weren't accurately modeled at all by functional MRI. And I think perhaps more concerning, the maps, while rich on both hemispheres, showed many asymmetries that are not convincingly neurobiologically robust, I would say. So in general,